Welcome, everybody. My name is Cliff Bohm, and I'd like to present to you the work I'm doing on my thesis. So, everything alive on Earth today is the result of a single process that has been running for over 3 billion years. You all know that already. Evolution by means of natural selection was first described by Darwin 165 years ago and has been formalized and extended by countless scientists ever since. In this talk, I will discuss some of my contributions to this work. In particular, I will share what I have learned about transient dynamics that arise in populations during periods of disequilibrium. Disequilibrium describes a state in which forces acting on a system create an imbalance, resulting in a system that changes over time. Sometimes disequilibrium is temporary. It begins when something disturbs a system and lasts until a new balance is reached. The disequilibrium can also be persistent. In this case, the system is always shifting. It's a stable disequilibrium. In these systems, the only constant is that nothing remains the same. Even in this case, however, there may be detectable, regular patterns of consistent change, and this is called a dynamic stable state. In a dynamic stable state, the system is constantly changing, but in a predictable way. Much of the work on evolution has not explicitly considered how evolution behaves during periods of disequilibrium. Instead, models have either focused on equilibrium conditions, so-called stable state models, or have considered trends over long enough timescales and over average behaviors. In such models, differences between equilibrium and disequilibrium may not be independently addressed. In addition, high-resolution modeling is hard. At least it's hard if you use the tools that have been the stock in trade of evolutionary research, which is mathematical formalizations, and empirical biological data. By hard, I don't mean it would be a lot of work. I don't mean that people aren't willing to do the work. I mean really hard, like intractable. These are uncalculatable systems. They are too complex, given our current technology. Get me some quantum computers, maybe we can do it. I believe that the reason why the work I'm presenting today is novel has more to do with the fact that I have access to computers. A lot of computers. Thank you, ICER. And I have access, and that allows me access to a different approach, and that approach is agent-based modeling. So where traditional models generally aim to build mathematical frameworks that make accurate predictions, agent-based modeling takes a different approach. Here, we model individual agents, and we observe how the actions of these agents generate system dynamics. Rather than deriving these mathematical models that fit observed data, I model populations of individuals. I observe their behavior, and I try to make sense of the dynamics that arise in terms of local interactions and global trends. Okay. Sorry, I can't see my next slide, so I don't know where I'm going. Agent-based models running on computers can simulate complex nonlinear dynamics that are analytically intractable. This is precisely what's needed to study the unstable disequilibrium conditions that interest me. I found that while disequilibrium states may be rare, they are a necessary condition that must, from time to time, arise during evolution. Moreover, during disequilibrium, the rules that describe the behavior of stable populations may deviate from existing models. But I'm getting ahead of myself, so let's start with some foundations. What is evolution? Evolution is a process by which diversity in life is generated. Evolution is a process of filtering where more fit organisms tend to outcompete less fit organisms. What does it mean to be fit? Well, it means to fit into the current environment. Fitness is relative. Whiter fur is beneficial for bears in the Arctic, not for bears in New Jersey. For individuals, fitness is simply the number of offspring produced by that individual. For populations, fitness is the average number of offspring produced by the population. A fitness greater than 1 indicates growth. while a fitness of less than 1 indicates decline. When the fitness is equal to 1, the population is stable, neither growing nor shrinking. What determines fitness? An organism's fitness is maximized when an organism is in sync with its environment. Perhaps not consciously, but at least structurally. Fit organisms have information or have access to mechanisms that allow them to acquire information. This information allows them to anticipate or predict what will happen and choose how to benefit or at least avoid harm. How does fitness change? Fitness can change in several ways. An environment may change, altering the value of an organism's information. Or an organism may change because of changes to their genome. What is a genome? The genome stores the information that an offspring inherits from their parents during replication. Technically, a genome is made of DNA, a structured molecule made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. DNA can be copied with high fidelity. DNA acts as a regulatory system that is involved in construction and operation of biological organisms. However, I will be using genome more loosely to refer to any element that an offspring can inherit. This includes non-genomic inheritance, such as immunities, culture, technology, and language. A fitness landscape is an abstract model that maps the relationship between an organism's genome and its fitness. On this landscape, altitude maps to fitness, and within this abstraction, changes to an organism's genome move it along the surface of a landscape. Fitness and landscape are simplifications of reality. A fitness seascape may be a more apt model, since the mapping between genome and fitness can be fluid. Landscapes shift over time as the environment or the population features change. And I'm happy to debate this point, but it's a nuance that is outside of the scope of this talk. So consider this simple one-dimensional fitness landscape. In this example, a single mutation can only move an organism slightly to the left or slightly to the right. 3 peaks are depicted, showing 3 local optima. These local optima are points in genomic space that correspond to local fitness high points, where further changes would require getting worse before one could get better. What is not shown is the actual effect of the mutation, the actual effect that mutations have on organisms. But for this, I'll give an example. Let us assume that the organisms on the landscape are snakes, and that changes to the genome depicted on our fitness landscape are the result of color changes from dark to light. It happens that in the snake's environment, there are a few dark rocks, some gray plants, and a lot of light-colored sand. When a snake's color matches the color of one of these features, they have a place to hide, making them a more effective hunter. In between these colors, though, they stand out and have a harder time hunting. Each peak in the landscape maps to the genomic configuration that correlates with one of the colors in the environment. The first peak, black skin color, only provides a small advantage because the dark rocks are rare. The middle peak, gray skin color, maps to higher fitness because gray plants are more plentiful. And the highest peak, the sand color, has the highest fitness because snakes of this color can hide almost anywhere. Let us assume that at some point in time, all of the snakes in a given area are gray. What would it take for snakes at this genomic configuration to move up here where they would be light-colored snakes? They would have to cross this valley. However, every mutation that would allow them to cross this valley will make them worse. Some lineage of snake would have to get very lucky over consecutive generations to accumulate the deleterious mutations that let them cross the valley. And for each additional mutation, the probability that snakes with that within that generation would survive and replicate would decrease. It would take a very long time to get that lucky. Thus, the population of gray snakes is in equilibrium. This is, there is a little opportunity for change because purifying selection, this selection against being worse, is strongly selected again, or purifying selection is strongly selecting against moves off of this gray peak. So how would things be different if the majority of the population were black snakes and there were just a few gray snakes? Perhaps the mutation for gray skin was just recently discovered in the population of black snakes. Now the population is not in equilibrium. There is a high fitness type that will, that is in English, there's a high fitness type that will produce more offspring and grow in number. I'm going to now replace these snakes, these images of snakes, with dots, and each dot will represent 100 individuals of a given genotype. So what we have is, we have a lot of black snakes. They're all clustered. Their genomes are all near this peak that represents having black skin. And then there's one snake, which is representing, say, 100 snakes in the population, some small percentage of the population, which is on this peak towards gray skin. But they're up the peak high enough that they're close enough to the color of the plants that they're already receiving an advantage. So I'm going to move this, I'm going to make this smaller. I'm going to add a couple features here. So I've added two features here. One, I've added this blue water line. The blue water line represents the average fitness of the population. So I talked about the fact that fitness can be on an individual, the number of offspring they can produce, or I said that fitness can be on the population level, it's the average. And what we see is that, note that these dots, the position of the dots don't matter. What matters is where the line is under the dots. And notice that that one guy who's up there climbing up the peak to gray, that Y peak, he's well above the waterline. The point on the line that correlates with his position is well above the waterline. This guy is really fit. He's going to produce more offspring. So he's going to grow in number over time. And the population is going to shift from everybody being black snakes to this sort of intermediate point where we've got a bunch of gray snakes. So something I want to point out is that we're still in disequilibrium. We haven't arrived at a stable state. The typical trend then would be that over time, because all of these guys on the X peak, the black skin peak, are below the water, they have fitness below average, they're reducing in numbers, they're going to die out. And eventually, we're going to just have a bunch of gray snakes. And then the system will be in equilibrium. But something else may happen. Something else may happen on the way to fixation, to stabilizing on that peak. Consider if the initial conditions are exactly the same. Some snake has just discovered gray skin. But then a happenstance, a mutation occurs that puts an organism on this slope up to the peak that would make them sand colored. This individual now has advantage over everybody else. But notice that the only reason that they could make this discovery is because the valley was still above the waterline. The average fitness was below the bottom of the valley. And so there's two possibilities here. We can see what happened in the top, where they failed to make the new discovery. And here, they did, and then we end up in the same state, right? This is the same state as here. They're just now shifting up to the new peak. So an evolving population can be in a fixed state, equilibrium, or it can be changing, disequilibrium. When in a state of disequilibrium, exploration and discovery are increased because advantaged individuals can afford to make accidents, they can afford to explore. Because disequilibrium populations are more likely to make discoveries, we say that these populations have adaptive momentum. And that's adaptive momentum. That's it. That's the whole thing. It's not that complicated. At this point, I could probably be done, but my timer says I have 30 more minutes. So I guess I'll talk some more. So you hopefully at this point, so how do we quantify this? How do we prove that this is the case? Chris said that they were working on this 15 years ago, but Avita was too noisy. So what we needed to do in order to nail this down, in order to say, look, here is what the effect biologically implausible. So first thing we're going to do is I have a 1D ring population. So every line here is one generation. I'm going to be showing you figures later. Every horizontal line is one generation. These three organisms are in this order. I should point over here so people online can see. So if I go up here and I say, here's one guy, here's the guy to his left, and here's the guy to his right, this order matters. Because when they reproduce, these three individuals in generation 1 will be competing over this location in generation 2. This is a spatial model. There are many reasons why we're using a spatial model. I'm not going to go into it at the moment. I'm happy to answer that in questions. And so we're using this 1D ring population. So the guy all the way on the right side is next to the guy on the left side. Just think of this thing as a cylinder. So we're sort of projecting down through time, down a cylinder. We've got these spatial competitions of size 3, and we're going to be varying the population size. And why we're going to do that will become obvious. This is our fitness function. So what we're saying is we have some set of repeating sawtooth valleys. So we have a step up, it's a big step up, and then we have a series of step down. We have 6 steps down into the valley, and then on the 7th step we go up. And this repeats infinitely in both directions. So it doesn't matter where you go on this function, your genome can either move you, or mutations to your genome can move you one position this way or one position this way. So if you're here, your offsprings can either be here, here, or here. And most of them, like more than 1 in 100, more than 99 in 100, will be exactly like you. The mutation rate is very small. So then we run a bunch of different experiments, and I'm going to explain to you some metrics that we collect. So the first metric we collect, don't be bothered by the names, it doesn't matter, it's just going to be TST. TST, or the blue one, is the stochastic tunneling time. So remember how I told you if we put all the snakes on the gray peak, and we say, okay, evolve, become sand-colored. It's really hard because they have to get across that valley. They have to find their way across the valley so that they can become sand-colored, and the valley is underwater. So the stochastic tunneling time is if we start all snakes gray and ask how long until, on average, until we get sand-colored snakes, that's the stochastic tunneling time. Then there's the neutral drift time. The neutral drift time says, well, let us assume that rather than having a valley between the gray snakes and the yellow snakes, it's the same width, it just doesn't go down. It's not harder, it's equally hard. And it's just a matter of random rolls. The way that you can think about stochastic tunneling time and neutral drift time is sort of like what they call a random walk. You're just rolling dice and you're saying, how long until I get a bunch of 6s? Stochastic tunneling time is like you're rolling dice, but you're asking, how long do I have to wait until I get 6 6s in a row? I have to keep rolling, and if I get a 6, I say good, 1. And if I get another 6, I say good, 1, 2. And then if I roll again and I get a 3, well, then I have to erase them and I have to start again. So that's the difference between the stochastic tunneling time that is against purifying selection, and in neutral drift, we've removed the purifying selection. The third thing is the fixation time. So on the diagrams that I showed, I said there are mostly black snakes and there's a few gray snakes. And we ask, how long does it take to go from a few or one gray snake and everybody else is black snake until all the black snakes are extinct and it's all gray snakes? That's fixation time. And then finally, there's the observed tunneling time. So TOBS, this is what we actually see when we run our experiments. So in a way, the stochastic tunneling time is a prediction. I'm saying, well, under a normal assumption, I have an expectation of how long it's going to take to make discoveries on this fitness landscape. And that's going to come from stochastic tunneling time. But then when we run neutral drift time, we're kind of asking what's the fastest that we would expect this to happen. If there was no purifying selection, if nothing was keeping these organisms on a peak, what's the fastest that we would expect? And then there's the observed time and the loosely, if you think about it, you say, well, I think the observed time is going to be like the stochastic tunneling time. Okay, so I'm going to put those metrics up on top just as a reminder. And now we're going to be looking at some data. It's going to get more technical from here on out, sorry. So on the y-axis, we have average times, and on the x-axis, we have population size. And it doesn't particularly matter, but note that these are both on log scales. So the first thing is we plot the fixation time. Now, since we have this 1D population, the fixation time is very related to the population size. It's a one-to-one relationship. As you increase the population size, it takes longer for the advantaged genotype to spread through the population. Then we're going to add the stochastic tunneling time. So this is the time that it takes to discover, if you're all gray snakes, how long until you discover sand color. And we see that decreases near linearly, not quite linearly, with population size. Then we look at neutral drift. Neutral drift does the same thing, it's just faster. So these crossing times are shorter, that means that they're faster, and they also decrease as a function of population size. I can tell you that if we go to infinity, because of the way that our mutational schema is set up, organisms can only mutate one position to the left or one position to the right at a time, and the value is 6 mutations wide. So these things at infinity are 6. In real biology, they would be 1, because any mutation on an infinite population will occur in the next generation. So now we add the observed line, and it does something unexpected. Well, okay, when we have small population size, the stochastic tunneling time predicts what we see. Okay, so far so good. And all the way at the other end, all the way at the large population, 10 to the 5, we see that the observed time flattens out, and it's taking longer than the expectation from stochastic tunneling. Okay, so I haven't been totally honest. We did expect this because there's something called clonal interference. If all the snakes are gray, and I have a huge population of gray snakes, multiple different parts of the population could be exploring, even though there's purifying selection, there's just so many of them that they're going to be making discoveries concurrently. The problem is that in the long run of evolution, because we're not just looking at a single run here, when we run the observed, if you look up here on the right, we're looking at not single steps like we are with stochastic tunneling and neutral drift, we're looking at many steps over time. We are making many discoveries here in this red line, but we're not mapping when we make discoveries, we're mapping which discoveries matter, which ones go onto fixation and become the seeds of the next discovery. And clonal interference, what's happening is that you have multiple discoveries happening concurrently and only one of them can win. And so you're losing, you're basically wasting money. You're getting these discoveries, but then you're not using them. So that we kind of expected this flattening out, but this is the interesting thing. In intermediate population sizes, the observed rate of adaptation is faster than we would have expected from stochastic tunneling. Okay, so what's going on here? Oh, there we go. So I'm going to add a marker here, B, and B represents a small population. It's around a population size of 200-ish. And what I'm now showing you is these additional 2 plots. So the plot in the middle is showing the distribution. These lines are showing the average times. But what I've now shown you is the distributions that make up those average times. And what we see is that not only are the red and blue lines on top of each other in the plot on the left, but in this plot in the middle, the distributions are on top of each other. The process that's generating these distributions is the same process. And why is this? Well, if we look at this thing on the right, what we see is individual discoveries. So each of these numbers, 0, 1, 2, 3, 4 along the left, those are individual discoveries within our trait. So those are individual discoveries stepping up our sawtooth. And what we see is that a discovery occurs, and then if you look at the little inset, the red line shows the population average. It fixes almost immediately. It fixes way before another discovery appears. So what happens in this region is we make a discovery and we immediately fix it. All right. Now, C represents intermediate population sizes. I think this is 2,400 individuals in the population. And this is what we get now. So the blue distribution looks very much the same. So the blue distribution is the stochastic tunneling time. And it looks quite the same. It's shorter, but we knew it was going to be shorter. But the red distribution is doing something very interesting, because the red distribution has two modes. It has a fast speed and it has a slow speed. And why is that? Well, if we look at the actual discoveries, so this is, again, we're just taking a little snippet out of a typical run. And if you look at the inset, it looks very much the same, right? We have some period of time and then we make a discovery in some period. But if you look, the heights of these discoveries, so this one was a single discovery here. But if you look here, we have 1, 2, 3, 4, 5, 6 in rapid succession. So what's happening is that the system goes into disequilibrium. It has an increased capacity to explore, and it's leveraging that increased capacity to explore. It makes another discovery. That keeps a window open, because now it maintains this equilibrium. As long as it maintains this equilibrium, it has enhanced capacity to explore. Eventually, it gets stuck. It lands at a point where it runs out of steam. So adaptive momentum can result in cascades of clustered beneficial mutations. There's an idea, I got enough time. There's an idea called punctuated equilibrium. These two guys, Eldridge and Gould, sort of were the main proponents of this. Unfortunately, they chose to attach it to some theory that ended up not working out so well. And everyone threw out the whole idea of punctuated equilibrium. And I'm telling you, punctuated equilibrium happens if the system is in the correct state. So let's look at this last point, D, which is a large population size. And this is just going to confirm our expectations. Again, we have a single mode distribution for stochastic tunneling time. But now we see an offset between the stochastic tunneling time and what we observe. And the offset is because of clonal interference. What this looks like here is that we keep making new discoveries. That's the black line. That's the leading edge, the current highest fitness. And the average never catches up. This population is in a stable disequilibrium. This is the important thing to note here. This looks, if I zoom out far enough, and in typical genetics models, you zoom out far enough, you think this is a stable state. And I'm telling you, this is a stable disequilibrium. This has as much to do with the top condition as it does with this middle condition, because we are in a constant state of disequilibrium. That means adaptive momentum is on the top. It's there, but it's so short. The windows are so short, it doesn't have an effect. In the middle, we see these cascades. We get a very obvious signal. Down here, it's in effect all the time. It's the norm. So now we can talk about something really neat. Typical theory defines regimes of evolutionary behavior. So the first regime I want to talk about is sequential fixation. Sequential fixation is the idea that the population size is low and the mutation rate is low. And so we accumulate a mutation and then it fixes. We accumulate a mutation and it fixes. There's another regime called the clonal interference regime. In the clonal interference regime, we are getting mutations so quickly that we can't fix them. And so we're constantly, the population is just constantly churning. There's just a lot of activity going on. And what we've observed is that we need to talk about a new regime, the balanced regime, which sits in between these two regimes. This, to me, may actually be the most important piece of this work, because this idea of evolutionary regimes is really central to theory of evolution. And adding a third regime, I think in the long run, this, if I had to put money down, this is maybe the bet that I would make as the most significant contribution. Or no one will use it and it's wrong, and that's fine too. So where does adaptive momentum matter? So adaptive momentum is an increase in the discovery resulting from disequilibrium. However, adaptive momentum does not always generate a clear signal. In the sequential fixation regime, fixation is too fast. In the clonal interference regime, the population is always in disequilibrium. So it's adaptive momentum is having an effect. It's changing the rate of discovery. It's reducing it, but you can't really tell because it's doing it all of the time. So adaptive momentum will generate the strongest signal of functional evolution when the fixation time is in between the neutral drift time and the stochastic tunneling time. So I haven't talked about neutral drift that much. The reason why we ended up plotting neutral drift was we said we came to understand what adaptive momentum is, and we came to understand that adaptive momentum is the relaxation of selection on advantaged individuals in a disequilibrium population. And we said, what's the fastest that we would expect them to go? Well, the fastest that we would expect them to go is if you relax selection entirely. If you relax selection entirely, that would be the neutral drift line. So we said, okay, the observed line has to live in between. In the balanced regime, the observed line has to live in between stochastic tunneling. It's not going to be longer than stochastic tunneling unless you get into clonal interference, but it cannot be faster than neutral drift. So we plotted neutral drift as, rather than trying to estimate where the time under disequilibrium is, we said, no, that's going to be an approximation. We don't want to do that, but we can give you this absolute number, this neutral drift number. So now I'm going to talk about something super complicated or super next level. So if you don't follow this part, it's A-okay. I'm going to talk about the notion of an over-dispersed molecular clock. So in population genetics, dispersion is used to measure the rate of the appearance of mutations in genetic histories. And it's some math, but basically you can count the number of events and you can use how these events are distributed in time. How regularly are they distributed? If you have a random event, again, if I'm rolling dice, I expect a certain distribution of outcomes. If every sixth roll exactly is a sixth, right? That's going to fit the distribution. I expect 1 in 6 rolls to be a sixth, but I do not expect the sixth roll to be a sixth every time. I don't expect to roll, something, 6, something, 6, something. That would be really weird. You would say, okay, I think something is up. So this is what variance, what dispersion tells us. It tells us how dispersed is the distribution. Dispersion or an R of 1 suggests that you have a random process, what's called a Poisson process. It means the thing is behaving as we would expect a random thing to behave. If the R is less than 1, it suggests a regular distribution. It suggests an overly regular distribution. That's the 6 is appearing on every sixth roll. And if we see greater than 1, it suggests clustering, which is the exact kind of clustering that we see when we have punctuated equilibrium. We see a whole bunch of discoveries very close together in time, and then these long periods. So here is the same lines, our stochastic tunneling, observed, fixation, neutral drift. And above this, I'm going to plot R for observed in red and stochastic tunneling in blue. So what stochastic tunneling does is it just becomes more regular, right? This R less than 1 means that it's becoming more regular. Why is that? Well, because in clonal interference, what you're doing is you're basically saying, I have a really big population. At small population, it's a random event. You're rolling 1 die. But now if I have 20 dice and I'm rolling all of them, and I'm asking how long until a certain configuration comes up, it's going to be more regular because I'm taking more samples. I'm basically taking the average of a process. And the observed line drops down for the exact same reason, because I have a larger population and I'm falling into clonal interference. In this case, I'm falling into clonal interference. The blue line can't have clonal interference because every time we make a discovery, we stop. right? We only record up to the first event, so you can't have two events that interfere with each other, but you still have a larger population, so you get this oversampling effect. But the interesting thing is that in the balanced regime, we see overdispersion. So we see that we get an irregularity that is more than we would expect, right? If it's below the line, it's too regular, more regular than we would expect. If it's above the line, it's weird. It's clustered, and that's exactly what we see. So we have evidence now on two lines, and this question of dispersion has been kind of a big question in population genetics because people have been using this to measure rates of adaptation. And here we have an incredibly simple system that explains how you can get dispersion. So visualizing adaptive momentum. So time along Y, we're looking at, oh God, I should know this. It's probably about 20,000 generations. Remember, each horizontal row, and they're really small here, is one generation. So when we see a triangle expanding, what we're seeing is a beneficial mutation, which we're going to show as a different shade of color, and then or a different hue of color. And then the different shades of the color, as they get darker, these are steps into a valley. And then when it hits the next lighter color, that's the next step to the next peak. So we're going to zoom in. Are we? Oh, sure. We're going to do this first. So I just identified a bunch of points that were interesting. So at the very top, we have what Vinny and I have called a self-actuated event. That is a discovery that the system made by random chance. This is like those gray snakes discovering the sand color. It took a long time. The system was in stasis for a long time before this. It makes the discovery. But once that happens, we go to a period of adaptive momentum. And you will notice that all of the discoveries are along leading edges. In a spatial population, the leading edge is where you have a fitness differential. On one side of the line, you have individuals with the benefit, on the other side without. So those individuals with the benefit along the leading edge can afford to explore genomic space. So we're going to zoom in right here. And we're going to zoom in right here. And what you can start to see more clearly here is these dark, starting here along the edge, we see the accumulation of deleterious mutations. And here we can see that the accumulation of deleterious mutations allows this to persist. Now, if this dark color was over here in the middle, purifying selection would clean it up. But because it's still part of the leading edge, it can explore across the valley. And you can see a light point here. You can see a light point here. These are discoveries across the valley, but they didn't fix. They didn't stick. They were lost. So I want to point out here also that in this image, every dot is one individual. So now we can actually see the actual individuals, right? So we're seeing this guy right here. There were three discoveries here, but that's one of the discoveries, and you can actually see how it propagates and spreads over time. Okay, so I'm going to try to be a little.
So that's a fairly large population size, right?
This was, I want to say this was.
These consecutive.
Yeah, I think this was 20. I think this is 2,400.
Yeah. So
it's not massive, but. So adaptive momentum across traits. This is my most recent work, and effectively, this is the same fitness function that I showed. What we do in this work is we add two additional traits. So we're going to call the trait that we had in the original work easy, and then we're going to have medium and hard. And the only difference is that we increase the width of the valley, and in doing so, we increase the depth. We go one step farther down each time. And now we have to show this not as a single depiction, but we have to show three depictions. This is the easy genome, this is the medium genome, and this is the hard genome. And we're looking at genomic values. The mapping between genomic values and fitness are one-to-one, so we can look at it either way. But we're looking at we're looking at genomic values, but they've been colored as they relate to the fitness function. So here in the last plot, it got darker when we went into the valley. Here it just gets lighter because we're looking at the genome value. So I'm going to zoom in on this top region to point out what's something interesting that's going on. So we have a discovery, self-actuated discovery in the easy trait, and there's a second discovery here. And now if we look at A, well, okay, A is over here. A is a discovery in the hard trait. I don't know if you can see it on this slide, but leading up to that discovery, you can see a lightening in color here. And that lightening in color is the accumulation of deleterious mutations in the hard trait in the individuals who are advantaged in the easy trait. Then there's a discovery in the hard trait. And then following that, we can see an accumulation of deleterious mutations in the medium trait, which eventually leads to a discovery in the medium trait. Following the discovery in the medium trait, we can see accumulation of deleterious mutations in the easy trait, in this mass here. So talk about that in a second. And then I just want to point this out because it's cool. This episode of adaptive momentum comes to an end here. The population goes back into equilibrium. It doesn't look like equilibrium, but if we look at what happens is the medium trait in this subset of the population is elevated because they have a higher version of the medium trait. They're on a higher peak on the medium trait. And for everybody else, these guys over here and these guys over here, for these guys here in the yellow, they're elevated on the easy trait. And so when this subpopulation, which is here, it doesn't look like anything special under the easy trait, and this subpopulation, which is these guys, meet, they have equal fitness. And this is a depiction of colloidal interference. At this point, both of these are still growing to the left and right because they're growing into lower fitness space. But where they meet in the middle, selection can't tell the difference between them, and that line's just going to drift. Eventually, it'll go one way or the other, but effectively, it's just going to drift for as long as it drifts. So the point is that ostensibly unrelated traits, that is, traits that have no functional interaction, still can interact. They interact because selection acts on the level of the organism, not on the level of the trait. So if you've got some trait easy in an organism and they increase in trait A, right, and their offspring are now going to have elevated trait A, they're just better. They can afford to explore. That means that trait B can afford to explore. Trait A could afford to explore, but trait B can too. That means the discovery may be in trait B and may be in trait C. It doesn't matter. Here's the first sort of take home. I had these three traits, easy, medium, and hard, right? So we've got easy, medium, and hard. And if I run them, if I make an experiment like the experiment we did in the first paper, where organisms just have one trait, and I run them and I say, what is the TST, the stochastic tunneling time, it wasn't even that. What is the TOBS? Sorry, mistake. What is the observed discovery time if I run these as individual traits? And what I see is that 26,000 generations, 500,000 generations, almost 6 million generations between discoveries on the hard trait. But now I put them all in the same organism. And now we see we see that the time for easy drops a bit, but the time for hard drops to 187,000 generations. So we're talking about not 5 times, we're talking almost 50 times, 25 times. So something else I want to talk about is how there's another piece we could do here, which is to say, okay, so we see that that putting these traits together changes the rate of discovery, but how are they connected? So what we did is, I'm not going to go into a lot of detail here, but if you look at A, B, and C here, these plots show for the easy trait, the medium trait, and the hard trait, shows whether or not that trait is in disequilibrium, if that trait is currently shifting in the population. And what we can do is correlate the state of the trait with discoveries. And what we find, I'm going to go a little bit faster because I'm pretty much out of time, but I'm going to take 10 more minutes anyway, is that the discoveries in the hard trait on their own, when nothing else is happening, if the easy trait and the medium trait are fixed, the rate of discovery in the hard trait is 6% of what we would expect by chance. If we just distributed those discoveries across time randomly, it is 6% as likely. And it is 5 times more likely, 5.3 times more likely, if the easy and medium trait are both currently shifting. So the adaptive momentum generated by the condition of the easy and medium trait providing advantage provides the cover needed to allow the hard trait to make discoveries at a much higher rate than we would expect by chance. Okay, so now I have a whole thing to still discuss. Sorry. The third paper that's in the thesis is predicting the unpredictable. This is using replay experiments to disentangle how evolutionary outcomes are altered by adaptive momentum. This was published at A-Life last year. So there are two pieces here. One is the adaptive momentum, which I think you guys understand at this point, hopefully. And the other one is replay experiments. So I have to talk about what a replay experiment is. So first of all, potentiation and replay experiments. This is the brainchild, or at least the thesis work of this guy here, Austin. And this is what he put in his thesis. And he looked over the wall between our cubes at one point and said, hey, can we apply my work to your work? And I said, hell yeah, we can. So really simple idea. If I put a basketball on this slope and I ask, how far down will it roll? Is it going to get stuck in that first dip? Is it going to get stuck here? Is it going to go off the end? And I can run this experiment a lot of times. So let's just zoom in on this first step here. So I put the ball here and I let it go. And I say, well, it's either going to roll down and end up here. It's going to end up here, or it's going to end up here. It's either going to get stuck in this valley or not. And this is like the stable state, right? And this is like making a second discovery. It's not a perfect analogy, but close enough. We're going to call, if it stays up there, fail, and we're going to call, if it goes over the hump, succeed. And then what we're going to do is we're going to take a single run and we're going to of a particular run. So we're going to take one experiment where the ball went down, and we're going to replicate it perfectly. You say, you can't do that. It's a ball and it's physics. And you say, oh, but we're in the computer, so I can. So what we're going to do is we're going to have a population of 512 individuals, and we're going to, there we go. We're going to put them on this fitness function. Blue indicates a first peak, and then this purple color is going to indicate a second peak, and we're going to take a small percentage of the population and put it on the second peak. Move these guys over here, and then we're going to plot the percentage of replicates that cross to this P3 peak, and we're going to look at where the leading edge is. So right now, the leading edge is at 0. It's all the way on the left. And so this is like the initial discovery. And then we're going to ask what percentage, and what we see is if we start with one guy or some small number of guys on P2, and then we run for a while, we have about a 18% chance that it will discover the next peak. So this was, Austin ran thousands of these experiments to get these percentages. These are like to the third decimal place. Then we say, well, what happens if we are at 100 individuals? Okay, so the likelihood goes down by a little bit. They have sort of less time to make the discovery. And what we see is, as we increase the population size, we see that the potential to cross. So this is so potentiation is the potential to discover a certain state. We arrive here, and this is very much like the intermediate state. About half the population is shifted over, and we see that the water line is coming up. So their potential to cross is reduced because they're running out of time. And the next point is lower and lower, and then right before fixation, the probability is basically 0. They're not going to do it. It's too late. They don't have enough time. So we can plot this, and then we can say, well, what happens if we start, rather than starting at P2, we start at P2 plus 1. So we start at this location, this one step over, and we see that our potential to cross increases by a little bit. Still, it decreases over time. We can go to the third step. We can go to the fourth step and the fifth step, and we can plot all of them. And so now we have empirically shown what is happening in adaptive momentum. The reason why I'm going over, I'm being very rude and I'm sorry about that, is that this work didn't do anything new for adaptive momentum. What this work did was say, no, we nailed the mechanism. This is what the mechanism is. So we have another one of these plots. Here we're going, time is going from left to right. Time is labeled on the bottom. And then the position in the population is vertical. Here are the colors representing the different genomic steps. And up here is the potentiation line. And the first thing I want to show you is that the prediction from the simplified model shows these strata, and the line matches the strata really well. And not only that, but if we drop, we see that we have this jump, this sudden jump in potentiation around 480 generations. And I'm zooming in on this yellow box, and I'm dropping where that yellow line is. And I don't know if you guys can see it, but there is a step right from here to here where we step from here to here. So it is literally that the leading edge discovers this point way down in the valley, one step away from the next peak. And that maps directly to the increase in potentiation. So I can show you some more of these, but I'm going to run through them. This is one where it fails to make the discovery. And you can see that the potentiation, it actually got above 50% at one point, but it runs out of steam. And then here's an interesting one. It actually discovers the next valley over. And what we see is that in the yellow here, the yellow is here. And so we're talking about now, we're talking about crossing this valley. Well, the yellow has to cross this, the P3 valley. And we can see that the potentiation in the yellow increases, and these increases, I haven't plotted it here, but they also map the same strata that were here. It's just that they're time offset because they would correlate with the point of 100%, which is up here. So, okay, so with that, I will make some concluding remarks. Adaptive momentum is an increase in the probability of valley crossing during disequilibrium events, such as selective sweeps and range expansions. Adaptive momentum is ubiquitous. It is a feature of all evolving systems. Selection efficacy is always reduced during disequilibrium insofar as it applies to advantaged individuals. So I haven't gone into detail on this, but we've talked about sweeps, and we've talked about spatial sweeps. In a spatial sweep, you have a leading edge. You have some individuals who have an advantage and the rest of the population who doesn't. A range expansion occurs when a population discovers new territory. This is the exact same phenomena. The new territory is just empty. There's nothing there. So it's like fitness zero. So everyone on the leading edge of the range expansion experiences adaptive momentum. In a well-mixed system, it's much more challenging, and we have some results that show adaptive momentum in well-mixed conditions, but it's much harder to show. What happens in these conditions is that a well-mixed population isn't structured, so you can't have a leading edge. What happens is that some individuals will have an advantage. Initially, one individual has the beneficial mutation, and then it will spread to more individuals. When one individual has the advantage, they have a big fitness advantage over everybody else. They're going to produce a lot of offspring. But over time, the average fitness is going to come up, and they're going to lose their advantage. This has some interesting consequences, because what it means is that it really makes clear the difference between absolute fitness and relative fitness. What it's saying is that the relative fitness of the new type, this genomic type, has a relative fitness advantage over everybody else, but over time it's going to fix. And when it does, its absolute fitness is greater than the old type. Its relative fitness, which is the actual number of offspring that it's producing, is going to drop back down to 1, because at some point it's going to be the entire population. It'll have no advantage. So even though it will always beat the old genotype is dead. And so it's not competing against them. So, and then the last thing is, imagine if you have a fish tank and you feed the fish a certain amount every day. And then at some point you just start feeding them more. Like from up till July, you feed them this amount. And then starting in August, you feed them more. And you see that the population in the tank grows. Well, the population in the tank is going to grow and stabilize at the new size. During that period, what this is called is a carrying capacity increase. You've increased the amount of food so they can support more fish in the tank. While the population of fish is growing, even though there is no advantaged group, everybody is the advantaged group, you still get adaptive momentum because you're going to have growth, which means that you can afford to take chances. And then selection efficacy is always reduced during disequilibrium, and populations will experience adaptive momentum as long as there are fitness valleys. If there are no fitness valleys, you're not going to experience adaptive momentum. You will experience a reduction in selection efficacy, but you won't experience an acceleration in the rate of adaptation. In fact, you'll experience a slowdown. So with this, I want to thank a bunch of people. First, I want to thank Austin and Charles, who worked with me on the A-Life paper, and ICER across the board for millions of hours of compute, and Beacon for getting me involved and training me on all this stuff. I want to thank these guys who have been working with me on the main paper, which we're farming out to journals as we speak. These two guys, as mentioned, here's a picture from, I don't know what, 94 or something.
93.
93, and here is us and Titus, and yeah, so there's me and there's these guys. It goes back. And I want to thank this guy. This is my partner in crime on adaptive momentum. I'm going to take this moment. We were sitting in offices and trying to figure this out. We were trying to figure out lots of different things, and it ended up being this. And actually, what Chris didn't tell you was that Chris and those guys figured this out. They weren't smart enough to figure out how to solve it, how to publish it. And then we accidentally discovered this because rediscovered it. We were trying to solve a different problem, and we came up with this thing. And Chris looks at it and goes, I think you guys just did something. You should read this draft that we were never able to publish.
Never even submitted it.
Yeah. So, and then this guy I want to thank. So Arn is the guy who hired me to make MABE, which then got me to go to conferences. And after like my third conference, and people would introduce me as Dr. Bohm, and I would say, no, my degree is just BS. And that joke got old. And I said, well, maybe I should just get the damn degree. So thank you, Arn. And then I want to thank my committee, who is basically everyone on the paper plus Louise, so thank you. And then I want to thank my family. So this is my brothers and sister and my folks and the nieces and nibblings and nephew. And I want to thank extended family. This has cousins, and there's no uncles in this picture, but there's cousins. And I want to particularly thank these three folks. So this is my mom. She's sitting over here. You can talk to her. This is my dad. He was a lawyer. He had his own set of very particular and very fixed morals and ethics. And I think that if anyone's debated with me about things and found me to be obstinate, it's probably inherited. And then this guy in the middle is my brother. He passed away a little bit ago. And I've dedicated the work to him. I'm not going to try to read this, but you can read it yourself. Was not expecting that. So, questions? Yeah.
So wonderful talk. Really, really awesome work. We're all often talking about how we should be looking at evolutionary theory for, the theory underlying evolutionary optimization and evolutionary algorithms. So can you comment on where you see adaptive momentum fitting in when we're thinking about how we think about optimization, solving optimization problems with evolution?
Yeah, there's like so many ways I can answer that. So in case anyone didn't hear, the question was, we can use these methods, the methods of evolution, to try to understand or to try to build, to try to engineer solutions. And this is called evolutionary computation. And the question is, how do we frame adaptive momentum in terms of this engineering endeavor of trying to leverage evolutionary processes to make discovery? And I think the first thing is, I think it's very difficult. I think that evolution is a messy process, and I think that adaptive momentum for me, my interest, so I have to be clear, my interest in studying evolution is how the damn thing works, and not how to leverage it. I'm sure that there are ways to leverage it, but I think that what this really does is a post hoc tool that allows you to see what happens. It allows you to understand how the thing works. How you leverage it is really hard, because really what's happening is we model random events. And what we're looking at is the distribution of outcomes based on random events. But what's a random event? What's actually random? Well, nothing, as far as we can tell, is actually random. There's an initial condition that's random. I set the random seed in my computer simulation, or there's some distribution of organisms and material in some part of the world, and then chemistry and physics happens. And so a big problem is we can talk about expected outcomes, but without knowing what the initial condition is and how the system is going to evolve. But I mean, in terms of the physics and chemistry, not in terms of the Darwinian evolution, it's the same thing. Then it's hard. But Chris wants to.
Yeah, well, I want to point out that you wrote a paper on how to apply that.
Okay, fine. So what we did, fine.
Where you
So
created basically a group of organisms that was on the side that
yes,
had no selective pressure and that was free to explore, and that would basically feed adaptations into the main population.
and what we found was that to optimize the size and duration that individuals should be protected from selection, you have to run a suite and probably have to spend all the compute time to find the right conditions so that you can leverage it. Vinny, do you want to say otherwise?
No.
Okay, anybody else? Yeah.
So for the multi-trait, it looked like there wasn't any synergy. Like, it's not better to have two traits that are under adaptive momentum for getting the third one to the peak. Is that true?
No.
Sorry, I asked a question about the middle section.
That's fine. You're good. You're good. I just have to get to the one plot. So this work, I will say, this is the newest work, and it has the least. That should be it. There it is. So this has the work. This has the least amount of work, which means I've thought about how to communicate this the least. What we're looking at on the bottom here is the background conditions. So none means all traits are currently fixed. Easy -- means that the easy trait is currently fixing. It means that there's some individuals at some level and some individuals at another level. Easy, medium, hard means all three traits are concurrently undergoing, like they're all shifting at the same time. If we look at the hard trait, so what these percentages are is if we look at all discoveries, we can compare discoveries to background condition. So background condition, for example, is only the easy trait is undergoing fixation, or all three traits are simultaneously undergoing fixation. What these percentages are is if we could say, okay, 60% of the time, in fact, I have this. Let's go back here. This is what I didn't have time to explain. So in the top plot, we're looking at the actual numbers of discoveries that occurred within each background. So these are counts of discoveries. There were quite a few. The next thing down is the ratio of discovery per background. So we're saying that 0.045 of all of the discoveries of the hard trait occurred in the none condition, and 19% happened in the easy medium condition, and 19.8%. And then we're looking at the background state ratio. We're saying how often was the population in each background state? So it's in the none state 73.9% of the time. It's in the easy medium 0.8% of the time. Right, but 19% of the discoveries on hard occurred in 0.8% of the time. So this map, these colors here, This is 5 times more likely than expected. This is 6% as likely as expected. But if only easy is going, it's only 3 times. If only medium is going, it's 3.6 times. If hard is going, it's actually 3 times, 3.7 times as well. But this ridge here is not by coincidence. The most likely condition for easy to go is when medium and hard is going. The most likely condition for medium to go is when easy and hard are going. And the reason why it's not here, the reason why it's not in this row is because there's a refractory period. After a trait makes a discovery, the nucleate, which is spreading, has everyone at the peak. None of them have explored into the valley. And so their ability to explore into the valley is not as high. It takes longer. They have to build up the population. Whereas the other traits could have already descended into the valley a couple steps. Does that?
Yeah, so there's lots of synergy.
There's tons of synergy. Yeah. Yes.
These peaks, they were the same like fitness benefit for the different traits, right?
Yes. Yeah. Everyone went up by 1 in this case. I mean,
So-
1.5.
I know there's a refractory period. Why do you think we see a difference between if we're looking at the hard trait, a difference between whether easy or medium is the one currently sweeping through the population? I don't see. So I'm seeing 3.1 versus 3.6.
I think it's because of the none condition, because easy can cross quite easily in the none condition. So the distribution, so I don't have it here. If I have 2 traits and they're both the same, then they're identical, but they're more likely to go when the other one is fixing. Right? So it's like they're a little bit less likely. But I think it has to do with how easy it can cross. I may not be right on this, but how easy they can cross under each condition. And because of that, their percentages are distributed differently. But that may not be. Do you have a different? So yeah, this is newer. So, all right. Any last questions? Yeah. Uh-oh.
So following up on an earlier question about lessons for people thinking about optimization kinds of problems, it seems like you sort of only at the end discuss spatial structure. But it seems like one lesson is that spatial structure is super important if you have any kind of valley crossing problem. So instead of it being sort of a limitation, that's actually a huge strength. And the trade-off would be if there isn't a valley to be crossed, but it's a hill climbing problem, spatial structure just slows everything down. So is that a fair statement of the fundamental issue? If there are valleys to be crossed, spatial structure helps you because of adaptive momentum. Otherwise, if it's a hill climbing problem, it probably slows you down because beneficial traits spread more slowly.
From a first approximation, yes. The issue is that it gets complicated when you start talking about different population structures. So hopefully this is some work that Alex and Austin and I are going to do, trying to figure out adaptive momentum on structured, like different structures, star structures, triangle structures, small world structures. The details will matter. But the way that I view this, it almost has more to do with the efficacy of selection and the amount by which purifying selection is limiting the capacity to explore. So, this comes down to the basic explore-exploit trade-off. If you maximize the strength of selection and you're on a kind, simple landscape, you're going to fly. And that's what you want to do. And in that case, having adaptive momentum is actually not going to help you at all, because it's just going to slow you down. It's going to mean during these periods of disequilibrium, you're going to reduce selection strength, which is going to increase exploration. But what are you exploring? There's nothing to explore, right? There's no benefit in exploring. And so, short answer, yes.
I mean, keep in mind that this is all controlled by the fixation time, right? So as long as you have a process that slows down fixation, it keeps the window open longer, and then that gives you, a better way to exploit adaptive momentum.
Only if there's values to cross.
Sure. But if there are values to cross, the lesson is by evolutionary computation, make sure things do not fix quickly. And if there's a way to prevent the fixation and keep the window open artificially, maybe that would be one way of.
Yeah, or just increase, or just create, I mean, what the Super Explorers paper did was create an artificial situation where we maintain disequilibrium on some percentage of the population, right? That was the idea, was to say, we'll just make this a permanent condition. Yep.
Addition to this conversation, because I'm saying there's this intuition that often will come up in conversation about just adding extra objectives, like making a problem multi-objective helps in the optimization setting. And this is what I can cite now when I want to make that statement. So I really appreciate it. Have you thought about adding like a simple optimization problem, maybe a PD optimization problem, and you add a couple of extra objectives that are just sawtooth functions?
Yes. Yeah. I mean, the original work that Vinny and I were actually, the questions we were actually asking had to do with what happens when you have multiple objectives. And there were a bunch of different hypotheses. One hypothesis is transfer learning, which is, oh, well, you learn something in one domain. You get good at playing chess, and then your brain understands grids, and so now you're better at playing checkers. Like you've never played checkers, but you sit down and because you have sort of spatial relationships, it all just makes sense. That would be transfer learning. But there was another idea that said, well, and this was kind of what I had in mind, but I hadn't sort of grokked adaptive momentum, so it didn't sit right, was, well, you can make trade-offs. So you're a sheep and you're optimized for eating grass, but they're also wolves, so you can optimize for avoiding wolves. So you might get to a local optimum on eating grass, but now you could get better at you could get worse at eating grass by getting better at avoiding wolves. But the problem was that in this model, it had to be a one-to-one trade-off. You could never go down. But what adaptive momentum says is actually there are windows where you can go down by a lot. And so certain values that would seem to be fully uncrossable during these periods become not just like, oh, you could get across that. They become like, it's actually kind of likely, in fact, in some cases.